What's the connection between this guy and this guy? Correct. So the cell will be busy making new proteins. It will be busy building new ribosomes. It will be busy making new mitochondria. It will be getting bigger. Lots of new proteins are being made. And what does a skin cell do? Well, it grows hair on it, a skin cell. So there will be, the DNA will be communicating to that ribosome, and they'll be making protein for growing hair. So that cell will be busy doing what it's supposed to do. Okay, so watch enzyme number 5 unzip. It's going to break those bonds between the G and C and A and T. Here it goes. All what our ancient C did. Okay. How about that for a during picture? That enzyme 5 is moving. You can see, and what were the words? What words would I put over here? Bond broken. Yes, excellent. Enzyme 5, before, and you can actually do these together, but it's also going to work on the AT DNA strand. And it's going to unzip that as well. So my during, whenever my hands are moving, that's our arrows, it's also going to unzip. Number 6 is going to pick up DNA nucleotides. So let's say it's going to pick up a G nucleotide, and watch what it's going to do. It's going to put that G right there. It's going to pick up another G nucleotide, and it's going to put that G right there. It's going to pick up another nucleotide, and it's going to put that G right there. Okay, what do you think it's going to do to the other side? Yes, it is. It's going to pick up a C, and it's going to put a C right there. I'm just going to move this up a little bit. Add a C here. It's going to pick up other nucleotide, another C. It's going to put it here. And et cetera, et cetera. And it's going to keep going, and I can just work on the C strand here. Number 6 is going to keep going until it adds all the nucleotides. It keeps these two pieces together, and it holds them together with this. It's, Spencer, you can tell me what this is? Good, and? Good. Protein and DNA, and it's going to hold, this is like it sticks these two new pieces that were made together. We have a name for that. We call that a centromere. We're going to add those DNA nucleotides that are T to the complement, to the A, and it's going to add to the T an A nucleotide. Okay? And it's going to keep adding those, just like I showed you with the GC, that I have a pre-built here. There's our AT, and another AT. Justin, what holds them together? Good job. So there'll be another centromere, protein and DNA. that'll hold these two guys together. The sister chromatid. So when the GC is duplicated, double, that is called a sister chromatid. When the AT is doubled, it's the same thing. So let's look at some of the language that we've covered so far. There's a lot of C words in this lesson. Originally, we started out with chromatin, which was our 46 DNA strands. Okay? When the DNA became doubled, the chromatin doubled, it was held together by a centromere. When DNA is doubled, it is now called chromatid. Okay, so in G2, we know this language, centrioles. The centrioles duplicate. Okay, so here we go. Centrioles here. They are going to duplicate, just like that. In your textbook, they have pictures that look like this for centrioles. So I'm just going to put those underneath. (3D) picture of our centrioles, microtubules. The second thing that happens, and I can already think about my Q column. Okay? These are steps. These are processes that happen within the cell cycle. The second thing that happens is the centrioles begin to grow these things. These things start to come out and grow from our centrioles. Yeah, well done. So the first stage in mitosis is called prophase. I got a model for you over here. Prophase. So this is what enzyme 7 does. I'll show you here what it looks like when DNA condenses. I need to pull this off the board to model this. This is happening on the chalkboard though. Okay? So enzyme number 7, my before picture here. During, watch what number 7 does. It condenses this sister chromatid, just like I rolled up that string. Okay, it condenses. Now I'm showing a rolling. It's actually a compression, almost idea, that happens with the DNA. Okay? Remember, this is a model, and we're just modeling for you what the condensing looks like. So I'm going to do the other. This would be happening at pretty well the same time. So my GC, my 1 GC is now condensed. At the same time, this other GC would also be condensing. So my before picture, GC, enzyme number 7 is going to condense that. And these are still held together by our centromere. Your notes, I'll do it up here. And we have another chromosome here. Okay? When your DNA is condensed, the cell can then do what it needs to do because it has to move it. That's perfect.
Dissolves. And that's the answer.
Perfect. Number 8. There we go. Nuclear membrane. The language that sometimes you see disassembles, breaks apart, don't like disappear. This is not a magic trick, right? The phospholipid bilayers are still actually in there. It's just broken apart. Okay? Did I say why? Okay, I want you to be thinking about why the heck would the nuclear membrane break apart? Ah.
Is it to allow the centrioles to like go in and then move the chromosomes?
Absolutely, you got it, good job. You look back on your microtubule lesson, spindle fibers move in a way. So they somehow had to get attached to them. Okay, okay, what's the next thing? Benjamin, we've got our next enzyme. Oh, that's okay, here we go, rock and roll, number 9. Number 9 is gonna break apart the cytoskeleton. Okay? Now, I'm gonna do something here. Can you chop, 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 chop? I am not chopping up my cytoskeleton. But I will kind of get it out of the way. Okay? So number 9, enzyme number 9 is gonna break apart our cytoskeleton. And attaching to those chromosomes. So they continue on growing their spindle fibers.
This is the 4th
centrioles.
stage of prophase.
This is all still prophase. Sorry guys, I forgot to put the label up here. And this is the end, this is what your picture looks like at the end of prophase. So what we're actually going to do is, I'm not going to do it, my spindle fiber's going to do it. They're going to move your chromosomes to the center of the cell, where we'll talk about that in metaphase, the spindle fibers are going to push and pull. And they're going to have the chromosomes lined up in the equator, which is going to be the center of our cell, just like the center of or the middle region of our Earth. Okay? So here we go. So this one's going to push these chromosomes to the equator. So this one's going to pull, and this one's going to push. Metaphase. Okay. Anaphase, which is the fastest stage, our next part in mitosis, this happens within a minute, seconds that this happens. I can't model it that fast. And some of you actually got to see this on your slides, which is awesome that you were able to even capture this. So the one on the left, the centriole on the left, with the spindle fibers, this spindle fiber is attached to this AT, and it's going to start to, it's going to pull, contracting here, it's going to pull that one AT. This one on this side is going to pull this AT. What we have happening here is one is going that way, and the other one is going the other way. What did I do to the sister chromatids? Yeah, and you got the right language out of B. The language is the sister chromatids separate in anaphase. Right up here, okay? So that spindle fiber is totally contracted into that centriole. This spindle fiber is totally contracted, whoops, to this centriole. Okay, the same thing's going to happen with my G and my C. And we have now separated our sister chromatids. Okay, questions, Brandon?
So in an actual human cell, would it be separating all 46 or 92, whatever it is.
Yeah, and I want you to think about something.
And then are there more centrioles, or is it just the two?
No. The last stage, anaphase. The last stage in our mitosis is telophase. Okay? And in telophase, the first thing that's going to happen is our chromatid, that's no longer a sister, it lost its sister. Where's its sister?
Separated.
Over there. Over there. Okay? It's going to uncondense. So our nuclear membrane is going to start reforming around these uncondensed pieces of DNA. And when that DNA uncondenses, because it's not doubled anymore, we're now back to chromatin. Okay? Did I have an 18 in GC when I started in G1? Way back? Did I have an 18 in GC when I started in G1 way back? Yes. The cell at this point, if you look at it in a real snapshot time, it can look like it has 2 nuclei. In telophase, we have our DNA that's uncondensed. We have our nuclear membrane that's reappeared. We have our cytoskeleton starting to grow. And the nucleolus reappears in the nucleus. And that is the end of telophase. And so the last part in the cell cycle, mitosis is complete. Those are the stages in mitosis. Our nucleus has been split, and we now have 2 nuclei in this single cell. So the last part is cytokinesis. And cytokinesis is really the splitting or division of our cytoplasm. Off the cell membrane and begins to pull that cell membrane in. Okay, I'll do it this side, and I'll show, I'll get another large piece of cytoskeleton here and show that pinching happening on the other side. Okay? And cytoskeleton that's found in this area will do the same thing. It will start pulling that cell membrane in like this. Okay, you can see it kind of, it's the fluidity of that cell membrane allows it to move and keep it in contact. So it's all still together. Okay. We call this little pinching in an animal cell a cell furrow. And the cytoskeleton continues to pull that cell membrane in. Pinching in, pinching in, pinching in. And you now have the production of 2 cells. And this, after cytokinesis, okay, that's the end of cytokinesis, your 1 cell has become 2. And each one of those cells will now return back into G1. So we have a G1 cell here, and we have another G1 cell here. And they'll go into the